Now let's look at the membrane potential. The neurons, their membrane potential is a little bit negative, minus 70 millivolt or minus 0.00007 volt. It's a little bit negative. And the reason is sodium-potassium pump. So let's look at the neuron. So that's the cell membrane. And the cell membrane, every living cell have the sodium-potassium pump. We talk about this in the unit 1. It used ATP as energy source. It pumps 3 sodium out. It pumps 2 potassium in. So every time it works, it pumps 3 positive charge out, only 2 positive charge in. So inside become more negative. And you also have the potassium and sodium leak channel. That's to prevent they overdo it. So they will pump more sodium in, more potassium out. But if they have way too much, they can go through the leak channel, so then leak out a little bit. So eventually the concentration inside is high sodium, but not too much. And outside is high potassium, but it won't overdo it. And they pump more positive charge out than in, so inside is a little bit negative. They put the electrode into the cell, they record the voltage, it's about minus 70 millivolt, and that's the resting potential. And the neuron, we call them excitable because they're able to generate action potential. So their voltage can go from minus 70 to positive. And let's see how they're able to do it. Apparently, they're able to open some ion channel. They're able to open the sodium channel and potassium channel. So let's look at how they do it. In the axon hillock, that's the axon hillock. That's where the action potential is generated. They have a lot of voltage gated because they sense the voltage. And voltage gated sodium and potassium channel. So when the neuron generate action potential, it will open the voltage gated sodium channel. And when the sodium channel open, sodium is high inside. Sodium is high outside, low inside. So sodium will quickly rush into the cell because it's high outside, low inside. So sodium rush in. When the sodium rush in, we're going to make the inside become positive. And we call this depolarization. So depolarization means the voltage go from minus 70 to positive. That's depolarization. And that's due to the sodium flow in. And sodium flow in causes the depolarization. Then the sodium channel close, potassium channel open. Potassium is high inside, low outside due to the sodium-potassium pump. So potassium is high inside, low outside, and you open the potassium channel, potassium gonna flow out. So potassium gonna flow out, and when the potassium flow out, inside become negative. They call the repolarization. So this whole process, this is one action potential. And the time from here to here, they call the refractory period. That's the time this neuron could not generate any action potential because these ion channels are busy generating this one. So in the neuron, the refractory period is very short. It takes about 3 milliseconds. So it can generate a lot of action potential. And that's why the neurons take a lot of ATP. And when we talk about the heart, we'll talk about the heart muscle have a very long refractory period. It's a protective mechanism. So it turns out your heart rate go to 150, 160, and maximally 200 beats per minute. That's the maximum because the heart have the long refractory period. So let's start the action potential. In the axon hillock, we open the sodium channel. So sodium gonna flow in. Sodium is high outside, low inside. Sodium flow in, cause the depolarization. And the sodium channel close, potassium channel open. When the potassium channel open, potassium gonna flow out. So that's the repolarization. And both channel close, that's one action potential. So the whole action potential takes less than 3 milliseconds. And this action potential will go through the whole axon until it reaches the axon terminal. So they can go it continuously. They call it continuous propagation. It happens in those unmyelinated axons. It's like you walk heel to toe, that's this one. And in the myelinated axon, they can use the saltatory propagation. This is like jumping. It can go faster. And that's why we need myelin. We need oligodendrocyte and Schwann cells create myelin because they can help the saltatory propagation. So that's this. In the central nervous system, we use the oligodendrocyte. In the peripheral nervous system, we use the Schwann cell. So they create the myelin, and they can create the saltatory conduction. So let's compare these two movements. Continuous movement in the unmyelinated axon. So the action potential starts from here, and this depolarization will open the ion channel next to it. And when this ion channel open, okay, sodium flow in, potassium flow out. Now the action potential go from here to here. It's like take one step, walk from with heel to toe. And this depolarization will open the ion channel around here. So now the action potential gonna go from here to here. So it's like one step at a time, walk heel to toe. For the short distance, this would be fine. But if you want to walk for a long distance, this is slow. So it happens in the unmyelinated axon. And let's see the myelinated axon. For the neuron, they decide to send the signal, travel for long distance, like the motor neuron, start from your spinal cord, go to control your muscle. This can be 1 meter long. And they need myelin. Myelin will cover over 90% of the axon. And between this we call the node of Ranvier, this is unmyelinated part, about 10% unmyelinated part. That's when the action potential happens. So the first action potential happened in the first node of Ranvier, and sodium flow in, potassium flow out, depolarization. Then the second action potential is not here because they are insulated by myelin. So the next one is actually here. So in the second node of Ranvier, they will open the sodium-potassium channel, then the sodium flow in, potassium flow out. That's the second action potential. Then the third one is not here, is third node of Ranvier. So the action potential can jump over 90% of the node of Ranvier. So they jump. It's much faster, especially when the signal need to travel for long distance. Those myelinated axons play a big role. So they use the saltatory conduction. And when you damage those myelin, you can cause multiple sclerosis. So eventually the signal may not be able to reach out. And that's why the multiple sclerosis impair the motor function. Even though all the muscle are normal, the problem happens in the neuron part. Okay, now the signal reached the synapse. So synapse is between cell and cell, between neuron and neuron, sometimes between the neuron and muscle. And that's the synapse. So, and there's a small gap. So in the gap, the electrical signal won't be able to pass through the gap because it's a physical gap. The gap is very small. It's nanometer range. It's 10 to minus 9 meter. So in the gap, in the synapse, the electrical signal need to be transferred into the chemical signal. That's the neurotransmitter release. So you heard a lot of different neurotransmitters like serotonin, acetylcholine, dopamines. These are all the neurotransmitters, and they all happens in the synapse. So that's where the synapse. So neurons, they receive a lot of input. On average, every neuron receives hundreds, if not thousands of input. And in the synapse, that's the synapse. The axon from the other neuron will touch the dendrite of this neuron. Sometimes they will touch the cell body, but most of them in the dendrite part. So let's zoom in to look at the synapse. And that's the synapse part. It's a small gap. So in the gap, the electrical signal won't be able to go through the gap. That's why the electrical signal need to turn into the chemical signal. That's the neurotransmitter. So apparently the electrical signal action potential goes through the whole axon terminal. In the axon terminal, it will trigger the neurotransmitter release. The neurotransmitter will pack in those vesicles. They use exocytosis. We talk about it in Unit 1. So those vesicles will fuse with the cell membrane, and the neurotransmitter inside will release out. And when it goes through the synaptic cleft, there's a very short distance. It's nanometer, 10 to minus 9 meter. And simple diffusion, this neurotransmitter will diffuse from the high concentration to the low concentration area, so simple diffusion. And in the postsynaptic dendrite part, they have a lot of receptor. And when we talk about the muscle, skeletal muscle, this part, that's the cell membrane of skeletal muscle, they have a lot of acetylcholine receptor for the motor neuron, because motor neuron use acetylcholine. So in the skeletal muscle, this part we actually call the motor end plate. So that's the muscle part you learned in previous chapter. But now we're in the neuron, so we call this postsynaptic dendrites. And this neurotransmitter will affect a lot of things, like serotonin tend to be associated with your mood. So people with low serotonin tend to be associated with depression and anxiety. So a lot of neurotransmitter affect the psychology and physiology. So the neurons, they will form the neural network. And this neural network, so far the scientists believe your memory, your emotion, all stored in the neural network, not in the neuron itself, it's the neural network. Okay, so this video shows you the cancer effect in the synapse. If you're interested, you can take a look. And this shows you the synapse, how they work. Okay, that's it.